Hello, everybody. I'd like to thank the organisers for inviting me to speak at this conference. And while my presentation is not directly related to COVID-19, it has important implications for vaccine development, antiviral immunity, and immune regulation in general. So the title of my talk is on natural killer cells as important regulation of human T-cell responses. Now, my name is Dr. Patricia Johnson, and I'm Principal Investigator of the Viral Immunology Laboratory at Dublin City University. So I'd like to give you some background to the Viral Immunology group at DCU. So here's where our laboratories are located on campus in Dublin City Centre. Now, the focus of the Viral Immunology group is to investigate immune responses to human respiratory viral infections. We're particularly interested in viral subversion of immune responses and induction of immune regulation. Now, the key viruses that we're interested in looking at are influenza, human parainfluenza virus, particularly type 3, and respiratory syncytial virus, RSV. The focus really is also looking at antiviral vaccine development for HPIV, RSV, and influenza. We have in the past worked with animal models of viral infection, but we are now developing a human ex vivo immune model based on isolation of human peripheral blood mononuclear cells. And this presentation will focus mainly on the results associated with this model. So the story really starts with human parainfluenza virus type 3. It's a single standard RNA virus. It's a negative sense virus compared to SARS-CoV-2, which is positive sense. It's a member of the paramyxovirus family. It causes respiratory illness in neonates and infants. Now, interestingly, there's currently no effective vaccine for it. And reinfections with this virus are common. So there's poor immunological memory. So the structure of the virus is that we have the nucleocapsid surrounded by an envelope here, and studded in through the envelope is our surface glycoproteins, HN or hemagglutinin neuraminidase, HN, or fusin protein, which is denoted F. So HN and F are the major surface glycoproteins of this virus. Now, to initiate a robust adaptive response, we need two key elements. We need to appropriately polarize our T helper cells, or our T cells, and we need clonal expansion of our lymphocytes. So this clonal expansion is key, particularly to the generation of antigen-specific memory cells. So if we look at HPIV infection, now animal models are poor models of HPIV infection. So in order to look more closely at this, we developed a human model in which we isolated antigen presenting cells or dendritic cells from humans. We infected them with HPIV-3, and we co-cultured these with allogeneic mixed lymphocytes. So this is an allogeneic mixed lymphocyte reaction. So this is a strong proliferative stimulus for lymphocytes. But despite this, in the PIV infected cultures, we had inhibited proliferation compared to infection with flu or treatment with TNF alpha, or even with dendritic cells or antigen presenting cells on their own. And this is we've seen it in our group, but this has also been published by other groups in relation to this virus. So HPIV inhibits lymphocyte proliferation. We also looked further, investigated this, and we found that there was reduced IL-2 in the cultures infected with HPIV-3. And we also noticed that the reduction in T cell proliferation was not due to cell death, but arrest of the T cells in the G0, G1 phase of cell cycle. And this has been published. So in order to look further at these responses, we purified out the CD3 fraction of the human lymphocyte population. And with this fraction, we saw robust proliferation of the CD3 purified fraction, suggesting that there may be another cell subtype within the mixed lymphocytes that's having a role in HPIV3 inhibition of T cell proliferation. So to try and identify which cell was involved, we directly infected the CD14, CD3 negative PBMC population with HPIV3, and we looked for the expression of functional markers and lineage markers. And we found that consistently the CD56 positive NK cells were increased in the HPIV infected cultures, but none of the other cell types were. So this suggested it may be the NK cell fraction that's having a role in the inhibition of T cell proliferation or lymphocyte proliferation associated with HPIV3. So the next group of experiments, we're looking at the NK fraction. We reconstituted the NK fraction in the purified CD3 co-cultures, and we saw that the NK cells, when the NK cells were present, that we could restore the proliferation, restore the inhibition of proliferation with the purified CD3s. Likewise, when we depleted the NK fraction from the lymphocyte populations, we restored the proliferation of the lymphocytes. So this strongly suggested that NK cells were having a role in the inhibition of lymphocyte proliferation by HPIV3. Now, we looked at the qualitative responses within the NK population. So there's two major subtypes of NK cells. There's CD56 bright NK cells and CD56 dim NK cells. But further studies identified that it was the CD56 bright NK cells that were responsible for inhibition of proliferation during HPIV3 infection. We also obtained virosomes from the virosomes containing the surface antigens F and HN from HPIV3. The virosomes are empty virus shells that just have these surface glycoproteins attached. And when we used these in the co-cultures, we also saw inhibition of T cell proliferation or lymphocyte proliferation compared to virosomes that contained surface glycoproteins from influenza or surface glycoproteins from sendivirus. So this suggested to us that these antigens from HPIV were important in the regulation of T cell proliferation. So in order to identify if either one of these are involved or both of them are involved in proliferation, in the inhibition of proliferation, we obtained antisera from Klaus Orville in the Karolinska Institute. So antisera from patients that have been infected with HPIV3. And what we did was we co-cultured the antigen-presenting cells, or DCs, with the anti-H. After infection, we added in anti-HN, anti-hemagglutinin neuraminidase antibody, and anti-fusion antibody, anti-F antibody. And we also had a control anti-distemper antibody to anti-distemper virus. And then we also looked then at whether this having antibodies to the viral antigens had any effect on proliferation. So in these cultures then, where we had anti-hemagglutinin neuraminidase antibody, we restored the proliferation, but not with antibodies to the fusion protein or our control anti-distemper virus. So this suggested to us that it was the hemagglutinin and neuraminidase component of the HPIV that was inducing the NK regulation of T cell proliferation. Now, a number of studies had been released suggesting that viral hemagglutinins, including hemagglutinin neuraminidase from HPIV3, bind to bind to NK receptors NKp44 and NKp46. So this suggested to us that these receptors on the NK cells may be involved in the regulation of proliferation, of T cell proliferation. So in these experiments, we blocked the NK receptor NKp44 and NKp46, and we also blocked NKp30, and we looked at this effect on proliferation. So whenever we had antibodies to NKp44, whenever we blocked P44 or the P46 receptor on the NK cells, but not the P30 receptor, we saw restoration of the inhibition of proliferation. And there was a cumulative effect when we blocked both receptors. And this happened in both the mixed lymphocyte populations and the purified CD3s cultured with our NK cells that had the various blocking antibodies. So these results were published in a number of studies, a number of publications, and they show us that the CD56 bright NK cells regulate T cell proliferation in HPIV infection by inducing a low level of IL-2 in the cultures, that anti-hemagglutinin neuraminidase restored the NK regulation of mixed lymphocytes and CD3 cells proliferation during HPIV3 infection, and that the regulation was occurred via the NK receptors NKp44 and NKp46. So that was interesting. But at the same time, coincidentally, with all these studies, a lot of reports came out suggesting that low-dose IL-2 has beneficial effects in autoimmune diseases. And these studies suggested that the beneficial effects were due to reduced T cells in circulation in these patients. Now, all of these studies suggested that while there was reduced conventional T cells and pathogenic T cells, there was an increase in T regulatory cells in response to low-dose IL-2, and also an increase in CD56 bright NK cells in these patients. And these were very consistent in numerous studies. Now, while a role has been suggested for the Tregs in the beneficial effects, little or no focus has taken place on the CD56 bright NK cells, or no role has been identified for these. But we thought that the similarity between these findings and our studies with HPIV suggested that they may have a role in controlling T cell proliferation and reducing T cell numbers in these patients. So we set forth to do a number of studies to investigate this. These have been published in the Journal of Clinical and Experimental Immunology last year. There's a number of experiments involved in this, and it's beyond the scope to go through each one in detail. What I'm going to do for you is give you a summary of the key findings of these studies. So what we did was we took out human peripheral blood mononuclear cells and we isolated the lymphocytes, and we subjected them to increasing levels of IL-2. And when we looked at this, we found that at low dose IL-2, similar to our HPIV studies, at low dose IL-2, we see elevated CD56 bright NK cells. And at high dose IL-2, we see elevated levels of CD56 bright NK cells. But these disappear from the culture at intermediate IL-2 levels. And this corresponded then with a reduction in lymphocyte proliferation of other lymphocytes within the culture at both low dose and high dose IL-2. Now, when we deplete the CD56 bright cells, we see a steady state of proliferation with the lymphocytes in response to doses of IL-2. So the CD56 bright cells were having this effect, reducing the number of lymphocytes at low dose IL-2 and reducing the number of lymphocytes at high dose IL-2, but allowing them to proliferate at intermediate levels of IL-2. Now, further qualitative studies suggested that the lymphocytes at low dose IL-2 were held in cell cycle arrest, rather like what we see with HPIV3. They were held, they weren't undergoing apoptosis. They were held in the G0, G1 phase. But at high dose IL-2, the T cells were being killed by apoptosis. This also corresponded to elevated cell cycle inhibitor P21 in the T cells, which corresponds with cell cycle arrest. And at high dose IL-2, we had elevated NK porphyrin, which corresponds to apoptosis of our lymphocytes. So these studies tied in very closely with what our HPIV studies showed. So then we wanted to investigate whether the receptors NKp44 and NKp46 were involved. So first of all, we looked at the expression of NKp44 and NKp46 with increasing levels of IL-2. And we saw that both of these receptors were present at low dose IL-2, and NKp44 was present in high levels at high dose IL-2. So we did the same blocking experiments with these antibodies. And again, we restored, if we blocked NKp44 and NKp46, we restored proliferation of the lymphocytes at low dose IL-2 and at high dose IL-2. In addition to this, we looked at the IL-2 receptor on the NK cells, and if we blocked IL-2 receptor beta on the NK cells alone or in combination with IL-2 receptor alpha on the NK cells, again, we restored the proliferation of these lymphocytes at low and high dose IL-2. So if we looked more qualitatively at the type of cells that were being, T cells that were being affected, markers of conventional T cells are reduced at low and high dose IL-2 in the presence of CD56 bright NK cells. And particularly the pathogenic ones, Th17, that can be associated with pathogenesis of autoimmune diseases. However, if we look at the Tregs, the markers of Tregs are elevated at low dose IL-2, which corresponds with the outcome in patients. We see elevated Tregs as well. So to look at whether these increase in Treg markers corresponded to increase in Treg proliferation, we looked at proliferation studies, and we can see that Tregs are again increased at low and high dose IL-2. However, they seem to be dependent on the presence of the CD56 NK cells for significant proliferation. And this is very important finding, suggesting that the NK cells are having an impact on the T regulatory cell proliferation. And also, if we deplete the Tregs from our cultures, we find the CD56 cells still have this regulatory effect on reducing T cell numbers at low and high dose IL-2. So the conclusions then of this study are that similar to our HPIV infection, the CD56 bright NK cells induce cell cycle arrest of conventional T cells in response to low dose IL-2 in vitro cultures of human primary lymphocytes. High levels of IL-2 result in CD56 bright dependent apoptosis of T cells. And the availability of both NKp44 and NKp46 receptors is necessary for cell cycle arrest at low dose IL-2. And blocking IL-2 receptor beta on the NK cells alone or in combination with IL-2 receptor alpha abrogated regulation of T cell proliferation at low or high dose IL-2. So Treg expansion at low dose IL-2 appears to be dependent on the presence of CD56 bright cells. So this suggests that CD56 bright NK cells have a role in the beneficial responses to IL-2, and they're somewhat overlooked in the immune regulation associated with this therapy. So these results then highlight the importance of NK cells in immune regulation and should be considered in human immune responses to vaccines or anti-inflammatory therapies. Now, one final thing I want to leave you with is also the fact that NKp44 is a human NK receptor which has only recently emerged in speciation, acquiring functional relevance only in non-human primates closest to Homo sapiens. So this mechanism of immunomodulation by the virus would not have been identified in any animal models of disease. So it's important that we focus on looking at human responses as well as animal models in relation to this particular immune regulation by NK cells. So I'd like to thank you again for inviting me to speak, and I'm happy to take any questions, but I'd like to first acknowledge all of the people responsible. This is a number of years of work, and a number of people have been responsible for this, and our collaborators from various institutes and our funders, the Wellcome Trust HRB and the European Union. So thank you again for your time and the invitation to speak.